Okay, here we go again. Sorry about this. I'm getting my exercise though. So I was just telling you about the fishy tomato. Okay, so in terms of genetically modified organisms. So if you have a tomato, well, I was saying that tomatoes grow in the Mediterranean and they grow in warm climates. You can grow them here. They don't grow very well near the South Pole or the North Pole. It's too cold. But what scientists have done is they've taken a gene from a type of cod, so a type of fish which lives in very cold North Atlantic waters, and it's thought to protect the fish from being exposed to the cold, to protect it from the cold, to give it cold resistance, in other words, because it lives in the very cold North Sea. And they decided that they were going to get the genome of the tomato, so they would have a cell and they would assess the entire genome, map the genome of that tomato, and then they would splice it. And after splicing it, they would insert, remember that just means slicing up the genome at a specific point, they would insert the gene which they took from the fish that lived in the cold water into the genome of the tomato that they're trying to grow. And then as a result, the aim was to grow tomatoes which are resistant to the cold as well, because if you can manage to get that DNA to replicate itself so that you can produce more and more and more cells that are the same, you could eventually grow a tomato plant. And this tomato plant now has a gene which codes for resistance to the cold, which the idea is so that you can grow it in a cold climate. It's very controversial, and I will say, as a pescatarian, I eat fish. I'm confused at whether it is part fish and part tomato. I haven't tried it and I wouldn't try it, but maybe you would. But the fishy tomato is a bit of an odd one, because we're not sure. If you've altered the genome, then it's no longer just tomato anymore, it's partly fish. Even though it's just one gene that's come from the fish, it's still partly fish genetically. So it's a little bit strange, and it's certainly not vegan. So that's gene splicing, and I will go over that in more detail a bit later on. And obviously it involves replicating the DNA, because every time you've spliced something, you've inserted a new gene, you then want to produce more and more cells of the same, which means you need to replicate the DNA each time. So number 3 is your DNA profiling and fingerprinting. So this is the forensics which we've already looked at. So if you're doing forensics, and you want to assess the DNA, you want to compare samples to other samples, then you might not have enough. So you might need to replicate the DNA so that you've got enough of it to work with, to compare to lots of different samples. So that's your forensics. It does involve replicating DNA in the laboratory. Number 4 is the same thing. You need enough DNA, so it's maternity and paternity testing. So usually paternity testing to work out who the father is of a child. So to do this, you need to have some DNA, a sample of DNA, and that sample might not be enough if you need to test it more than once against another sample, against the child sample. So you might need to replicate it many times in the laboratory so that you can test it accurately against the child. So paternity and maternity testing. And also genetic lineage, which just means tracing your ancestry. So when we do that, we look at your DNA and we compare it with people all over the world, and ancient populations as well, to see if we can work out where our ancestors came from, which can be quite interesting and surprising and shocking to some people as well. I have a Burundian friend that found out that he's not really Burundian at all, even though his parents are. Originally his ancestors are from a long, long way away. But to do that, obviously, you're going to need a lot of DNA. So one sample might not provide enough of the DNA for you to compare it with all of these different populations and samples that you have. So you might need to replicate that DNA in the laboratory so that you have enough of it to compare. Okay? And number 5 is genetic counselling of parents to inform them about inheritable disorders or conditions. So this is basically if scientists don't know enough about the condition and they want to replicate the cells in the lab. So you need to replicate the DNA so that you can grow more and more and more of these cells to to see what the effect is on the person, or what the effect will be with the condition, so that you can help the parents understand the inheritable disorder. You will need to do that. But